In this paper from the Laub lab at MIT, published in 2022, the authors are looking at specific versus non-specific compensation in co-evolving protein complexes. They're evaluating this to see the effect on the potential mutational space that the co-evolving protein complex can take. To study this, they're using an antitoxin and toxin binding complex. They first make libraries of all possible variants of the antitoxin and all possible variants of the toxin. They then do random combinations of these antitoxin-toxin pairs in cells and measure the cell growth over time. Functional toxin-antitoxin pairs will proliferate, while non-functional toxin-antitoxin pairs will die, leading to positive and negative relative growth rates compared to the wild type. They use this experimental setup to determine non-functional antitoxins, as well as binding and non-binding toxins. Ultimately, they use this experimental setup to determine which of the non-functional antitoxins can be rescued by variants from the toxin library. They call these toxin mutants enabling mutations. There are two different types of enabling mutations, nonspecific and specific. Nonspecific refers to enabling mutations that rescue all of the non-functional antitoxin mutants, shown in orange on this graph. Specific enabling mutations only rescue some of the antitoxin's functions, shown in purple. When they looked at the location of these toxin mutations compared to the mutation in the antitoxin, the specific enabling mutations were all very localized to where the antitoxin mutations, suggesting a direct compensation, whereas the non-specific enabling mutations are much more diffused and sometimes not even close to the interface of the proteins. So then, looking at the effects of these non-specific enabling mutations on the neutralization capabilities of the antitoxin and the antitoxin mutational space, the authors created combinatorial libraries of 10 of the most effective non-specific enabling toxin mutations with an antitoxin combinatorial library of 3 of the most common non-functioning antitoxin variants. And what they found is that these non-specific enabling toxin mutations were able to rescue more and more of the antitoxin functions, thus expanding the antitoxin evolutionary space. They also visualize this in the mutational graph shown on the right, where starting from the wild type antitoxin, the evolutionary space the antitoxin can take up with just the wild type toxin is clearly more limited than with the toxin within the non-specific enabling mutation. Also of note is the expanded function for the antitoxin to bind to new toxins. They studied still in the same family, but still expanding the function of this antitoxin. So why is this interesting? By studying this, perhaps we can apply these non-specific enabling mutations in antibodies to better tolerate evolving target antigens. Thank you.